Good evening, everyone. I hope you're all doing well and staying safe during this pandemic. I'm Monica AH, second year student of MS Farm in Natural Products, and I welcome you all on behalf of the Department of Natural Products, NIPER SAS Nagar, to today's virtual alumni talk on natural products driven discovery of new leads for modern medicine, sharing the learning and experience of translational research presented by Dr. Sandeep B. Barate, Principal Scientist of Natural Products and Medicinal Chemistry, Division at Indian Institute of Integrative Medicine, IIM, the constituent laboratory of Council of Scientific and Industrial Research before their cameras. Queries can be put forward in chat box, both in Google Meet and YouTube throughout the entire webinar. Dear participants, now let me introduce to you all the Department of Natural Products. The department has been involved in research activities since the inception of the National Institute of Pharmaceutical Education and Research, NIPER SAS Nagar. NIPER, being the Institute of National Importance, is an autonomous body set up under the aegis of the Department of Pharmaceuticals, Ministry of Chemicals and Fertilizers, Government of India. The Institute is conceived to provide leadership in pharmaceutical sciences and related areas, not only within the country, but also to the countries in Southeast Asia, South Asia, and Africa. The Department of Natural Products is one of the 6 core departments in NIPER Mohali, which encompasses a range of innovative research projects towards drug design and development of model bioactive compounds and new potential drugs or herbal formulations. The expertise of the department includes extraction, isolation, synthesis, and structure elucidation of a wide range of natural products, their standardization, development of different dosage forms, including traditional medicine formulations, nutraceuticals, and phytopharmaceuticals. Participants, today's guest, PhD alumnus of batch 2007, is Dr. Sandeep B. Barate, who will share his learnings and experience on natural products driven discovery of new leads for modern medicine. I now request Professor Inderpal Singh sir to introduce the speaker to the audience. Over to you, sir.
Good afternoon, sir. Sir, you are on mute. Sir, you are on mute. Yeah. Okay, sir.
Yeah, welcome, Sandeep.
Yeah, thank you,
It's
sir.
very good to see you here on this platform. So
Yes.
It's my pleasure to introduce Sandeep Barate. He has been one of the most illustrious students of our Department of Natural Products at NIPER Mohali. And Sandeep was my first PhD student when I joined here at NIPER. So I think I have seen him growing and he has seen me also. So I'll formally introduce now Sandeep. Dr. Sandeep is currently Principal Scientist and Head of the Department at Natural Products and Medicinal Chemistry Division at IIM Jammu, that is Institute of Integrative Medicine, Jammu, and Associate Professor in Chemical Sciences at Academies SIR. So currently he's at CSIR IIM Jammu. He did his PhD on fluoroglucinol compounds in 2007 at NIPER Mohali, and MS Pharm again at NIPER Mohali in 2002 under guidance of Professor Cacao Permeability. And he did his B Pharm from MDM VPS College of Pharmacy at Nasik. So his main research areas are medicinal chemistry for cancer and Alzheimer's disease, and also is working on natural products, phytopharmaceuticals, and nutraceuticals. So he has a lot of industrial experience also. He has worked at Dr. Reddy's laboratories in Hyderabad, Piramal Life Sciences in Mumbai, and then he has a postdoctoral fellowship from University of Montana in USA from 2002 to 2010. And since 2011, he is at CSIR, Indian Institute of Integrative Medicine. So he has very impressive publication record, 147 publications, 17 patents, and he has also out-licensed. His work has been out-licensed to industry. Several molecules like IIM 290, 141, and 160 as NCEs or nutraceuticals for pancreatic cancer, Alzheimer's disease, and rheumatoid arthritis. He has also received several awards and honors, including CSIR Technology Award, ACS Medi Young Scientist Award, OPPI Young Scientist, CSIR Young Scientist, NASI Young Scientist. So all possible awards for young scientists in India he has received. He has guided 11 PhD students and 7 are continuing. So again, very impressive. And he has received a number of research grants, 20 in number, out of which 9 have been as completed as PI and 6 grants are ongoing. So these grants have been from CSIR, DBT, DST, and other external funding agencies. So currently, he is at IIM Jammu, and he has been doing very good work there. And my best wishes to Sandeep for his future. And with this introduction, I would like to invite Sandeep to deliver his talk.
Okay, sir. Thank you very much for some detailed introduction. So it is always great to deliver any lecture for Naipur Mohali because it is I'm always feel privileged to be an alumni of IIII and Naipur Mohali, and I'm of this prestigious institution, and particularly the faculty, Dr. I.P. Singh and Dr. Jachak. So not only PhD but masters as well as PhD alumni. So it is always good feeling that I'm presenting to my alumni juniors and sharing my experience, how I learned my basic natural product research, structural illustration. Although during my masters I isolated very known natural products, but those learnings helped me to establish my research career and how I was able to translate those learnings to the products or clinical candidates. So those learning obviously started from Naipur and then how I translated in my independent research. So that I'll briefly present you in next 1 hour. So I'll share my screen here. I can see some of my colleagues, Dr. Siddheshwar is here. Atul is also here, I think. Atul.
Yeah. Hello, Siddheshwar. Good afternoon.
Hello, good afternoon. I'm very happy to see you all here.
Hello, Dr. I.P. Singh and Dr. Siddheshwar, Dr.
Hello,
Rohitokin.
Yeah, Raju is also here.
Hello, Raju. Hello, Raju.
How are you all? And it's very nice to see you all on screen. Unfortunately, my laptop camera is not working. Otherwise, I would have on my camera. So it is great to see you all.
Yeah. Hello, Atul. If you are listening.
Yes,
Okay.
Sandeep.
Okay.
Good afternoon, sir. Good afternoon, sir. Yeah.
Good afternoon, all of our alumni. Good afternoon.
Thank you, sir. So, can I take for 40 to 50 minutes around for lecture?
Yes, please, Sandeep.
Okay, sir. Okay.
Yeah.
Yeah. Can you see my screen?
Yes.
Okay. So, very good afternoon to all the students, my colleagues and faculty members. So, I'll be speaking about my research efforts which I did in last 10 years or 11 years in IIIM Jammu. So topic of my today's lecture is natural products driven discovery of leads for model medicine. So here I'll be sharing my learnings and some of the experience of how I translated my phytochemical investigation research into the products or some of the preclinical candidates or clinical candidates. So although there was a brief introduction, just I put my slide, my background of the speaker, so I'm not speaking about this. So both master's PhD I completed from Naipur Mohali. Sir, is it slideshow now?
Yes.
Yeah.
Okay. Sir. So briefly about me, after completing masters and PhD from Naipur Mohali, I work in industry for around 1.5 years in Nicholas Piramal, and then I went for postdoctoral studies in University of Montana. I gained interest in working on Alzheimer's disease, so that I started some of the work in IIIM on Alzheimer's disease. And during postdoc, always you get an opportunity to learn some of the not only chemistry, but you learn to do some classes, handle the cell lines, and those kind of experience for the first time learning during postdoc. And some of those I carried forward here, and I did some of the 2-3 PhD students also they did PhD in Alzheimer's disease. And since 2011, I'm working in IIIM Jammu at various positions in natural products and medicinal chemistry division. So before I go to my actual presentation of my own research, briefly I will show you what I did in Naipur Mohali as an alumni. So I did my master's MS Pharm in 2001 to 2002 in Naipur Mohali, and my supervisor was Professor Cacao Permeability. So this project that time we used to work under a PhD scholar. So that time my actual immediate mentor was Dr. Hema. So I was working on isolation of the for identification of various Osimum species, and one of the plant which was allotted to me as Osimum pilmans. Although here our objective was not to look for the new NC, new natural product, but our main purpose here was to gain the training on isolation techniques, structural elucidation, those kind of techniques was the main purpose. And during that exercise, in that time our project was up to only for 6 months. So during the period of 6 months, I could isolate 9 compounds. And these are the compounds. You can see most of the structures are very trivial, and they are in most of 57 when CSIR established in somewhere around 1950s. So this one of the laboratory was taken by the CSIR, and the name was translated to Regional Research Laboratory in 1957. And afterwards, in 2007, we led the mandate of the institute integrating the traditional knowledge with the modern medicine. So based upon that mandate of the institute, the name was renamed as Indian Institute of Integrative Medicine in 2007. So core components of this institute is that we have all sort of divisions like that, starting from the biodiversity, then plant collection, taxonomists are there, natural chemistry, medicinal chemistry, pharmacology, microbiology, fermentation technology, all sort of divisions are there. And ultimate aim is to discover something on preclinical leads based upon natural sources. So that is our vision or mandate of the institute. We have main campus in Jammu, which is established in 1941. And this is one of the CSR laboratory out of these 38 national laboratories under the CSIR. And we have another sister laboratory in Srinagar, which is established in 1971 and inaugurated by Vice President in 1971. Now I'll start my presentation of my independent research career at IIIM Jammu. So this is briefly about my contents. I will not speak much about because I cannot, within 1 hour, I cannot present each and everything in my 1 hour lecture. So I decided, since mostly my intention was to show that how natural product start working on a particular plant, how we can translate that initial basic research of isolating the natural product into the products or in clinical candidates or preclinical candidates. So these 3 important case studies I will be showing here. So these are the 3 plants where we work extensively. Almost since last 8 to 9 years, I have been working on these 3 plants, and we could deliver something in all these 3 plants. Although we could not isolate many new natural products, but that was our never intention. Our intention was to translate something into product, so we can out-license some of the product or something we can take to the clinical trial. So that was our main objective here. So therapeutic areas also I mentioned, like one of the lead we from this diazoxazolum we translated for oncology, then Crocus sativus for Alzheimer's, and Virginia acidata for rheumatoid arthritis. So these three plants I will discuss in my today's talk. So as far as the key objectives of my research laboratory, so these are the 5 important areas where I work, and each of them are interconnected to each other. So first is very important is a phytochemical investigation of some of the important medicinal plants. And then once we isolate some of the natural product, we look for any important natural product where we can do the semisynthetic studies, or we can go for the total synthesis. Some of those hits we select for medicinal chemistry program. And here I decided to work on oncology and some of the CNSs. And here I built my capabilities of even doing the computational studies. I have my own docking softwares and everything, workstations where I can design my own molecules for synthetic chemistry and medicinal chemistry. And also some of the in vitro assays, because while doing the medicinal chemistry, the major hurdle or maybe bottleneck is getting in vitro biology for SAR optimization in a quick time. And if that is not possible, then it is very frustrating to do the medicinal chemistry. So that is the reason I established some of the in vitro enzyme-based assays within the group so that we can drive our medicinal chemistry program at a proper pace. And obviously, while doing the medicinal chemistry, always we look for some of the important synthetic methodologies for medicinally important compounds. And second pillar for the phytochemical investigation is like look for the potential opportunities for translating those extracts or maybe enriched fraction as a phytopharmaceutical lead or even as a nutraceutical leads. So these are the 2 main pillars: medicinal chemistry and phytopharmaceutical leads. And ultimate aim in all these is to do some preclinical development so that we can reach at least up to the IND filing and plan some phase 1 proof of concept in human beings. So this is overall objective of my laboratory. So briefly, in last 10 years, these are the plants where we did some phytochemical investigation work. So just I summarize here, I will not speak about all the plants. So diazoxazolum, Virginia acidata, Crocus sativus. Then apart from these 3, then we also work on Malatus philippensis for some of the, here objective was to isolate some compounds and screen for cancer program. Then Actinocarya, Pongamia pinnata, Glyceraza glabra. And there is one more plant like Boira diffusa. Here also we work on this plant, and one of the natural products which we isolated here, Boiron B. This we identified as a very important compound which can reverse the MDR reversal, SRS MDR. And when we combine with some of the antibiotic drugs, it has reversed the drug resistance which was developed by this bacteria. So, but out of these, wherever we found the potential, those 3 plants we selected, and these 3 plants I will be discussing in this lecture now. These are the 3 plants now. Why we selected this plant now? Just briefly, I will show you here. The first plant is the Diazoxazolum binactifera. It belongs to the family of Melaceae. This plant, we can say it is Indian medicinal plant. This occurs only in India. So this is one of the beauty, and that was one of the reasons I was more interested in working on this plant. And this plant is a very huge resource for isolation of one of the unique natural products, the Rohitokin. This natural product is present in abundance in this natural product, and it has already inspired the discovery of 2 clinical candidates, 2 anti-cancer clinical candidates. This, based upon the structure, there are 2 candidates which have gone to the clinical trials, and multiple clinical trials happened. One of the drugs is already approved by the US FDA as an orphan drug. So this is one of the inspiration that Indian medicinal plant also can deliver the drugs for the modern medicine. So we wanted to put our foot in this area and to discover something out of the box, whatever is available in the clinical trials. Second plant is a Virginia acidata. Again, this plant, why we selected this plant already is available in multiple formulations which are available in the market. Like one of the plant formulation from the Himalaya drugs, they have one formulation in the market for kidney stones. And this natural product is present in abundance in this particular plant. Berlinin is the name of the natural product, which we can isolate in 1 or 2 gram, 2% from this plant. So that is considered as a very good quantity in the natural product. If you isolate more than 1%, it's considered to be very, very good. And another third point is it occurs primarily in the temperate Himalaya. So we can get this plant from the nearby places like Patnitop and nearby places. This plant is available. So we wanted to work on this. Third plant is a Crocus sativus. This is, I think, everyone knows saffron. Everyone knows saffron. Saffron, this is being consumed since very ancient times. A lot of traditional knowledge is there. It has been reported to have memory enhancing properties, and it contains unique secondary metabolites, carotenoid class of compounds, and it occurs in the Kashmir. Again, because of the location of this institute, we wanted to put our effort on this plant, which occurs in this Jammu and Kashmir region. So I will speak about all these three plants during my lectures. But apart from these three plants, the therapeutic areas which my group work is oncology and Alzheimer disease. So based upon these therapeutic areas, I have distributed my slides in these two therapeutic areas. First is oncology, where our efforts are on the kinase inhibitors. And the two kinase inhibitors which we selected is the CDKs and PI3Ks. And here our objective is to look for some isoform selective inhibitors and discover some of the preclinical leads. And Alzheimer disease, again, we are looking for the multi-targeted agents. So that I'll discuss in my coming slides. I will not speak much on this particular slide. So I'll start with the oncology, what are efforts on the oncology. So why we are working on this particular kinase inhibitors? So this slide will explain you. So oncology, these kinases, if you look at the kinases are used as a therapeutic targets or drug discovery program by multinational companies. More than 30% of the targets are kinases by major multinational companies, because kinases are implicated in majority of the diseases, and major contribution of the kinases or involvement of kinases in oncology. And these are the list of the drugs which are approved till date. The very first drug was approved is imatinib in 2001. It is EGFR kinase inhibitor and approved for the breast cancer. And after that, you can see the portfolio. There are almost now 65 kinase inhibitors are approved till date for various types of the cancer. And this particular slide is from our recent review, which is published in Medicinal Research Review. And if you see the distribution of the kinase inhibitor for various therapeutic areas, mainly contribution is in oncology, but there are some of the kinase inhibitor like in rheumatoid arthritis also there are 3 kinase inhibitors. In other diseases there are few kinase inhibitors are approved. So if you look at the CDK inhibitor, where we are more interested in, there are 3 kinase CDK inhibitors which have received the approval. So you can see 2015 first CDK inhibitor was approved by Pfizer. It's the palbociclib. It is a CDK4 and 6 inhibitor which is approved for the breast cancer. And followed by that, there are 2 more CDK inhibitors are approved, ribociclib and abemaciclib. And again, both these are for breast cancer. Another kinase which selected is a PI3K inhibitor. Again, this is a clinically validated target because there are 4 drugs already approved by the FDA. Because whatever target we select, it must be clinically validated. Then only it is worth putting a lot of money and a lot of efforts on this kind of molecular targets. So I'll start with how we started a Medichem program on these kinase inhibitors. So for CDK, we selected natural product scaffold. Our intention was to select some of the natural product scaffold and get something out of this natural product. So these are the 3 scaffold which we selected. First is a fascaplycin. If you see, this is a beautiful natural product, highly planar in structure, pentacyclic structure, and it is extremely potent anti-cancer compound. And it was discovered during the NCI screen during 1990s by Novartis. And this compound was reported as a highly selective CDK4 inhibitor. There are more than 12 to 13 CDKs, but it is inhibiting only CDK4. So there is something is available, something is there which is highly selective for CDK4 inhibitor. So that's why we wanted to work on this natural product. Another natural product we selected is a Rohitokin, which I spoke just now a few minutes ago, that this natural product has delivered 2 clinical candidates, and this natural product occurs in an Indian medicinal plant. So we wanted to do medicinal chemistry on this. And third natural product is again from the marine natural product is a merid-NNC. So we wanted to synthesize some of the new derivatives out of this. We did the total synthesis of this, and then we synthesized a couple of analogs and screened for different kinases. Another program was a PI3K, where our main intention was to gain isoform selective inhibitor, because in cancer, if you look at the PI3K, phosphoinositide 3 kinase. Here there are 4 isoforms of PI3K available in our body. Out of that, only alpha isoform is highly overexpressed in the cancer cells. So, and if you inhibit the other isoforms, there are chances of getting a side effect. So our main intention was to get the selectivity for alpha isoform. So from the basic research point of view, we wanted to select 2 scaffolds where we can, using the computational approach, we can discover something. Both the scaffolds are pan-PI3K inhibitors. Pan means they are inhibiting all 4 isoforms of the PI3K. But we wanted to discover something which will inhibit only alpha isoform. So from the basic research point of view, we selected these 2 scaffolds, and 2 PhD students work on each of the scaffold, and we could discuss something clinical candidates which are highly selective for alpha isoform. So briefly, I will talk about these 2 scaffolds also. So as outcome of our Medichem program on cancer, on kinase inhibitor, these are the kinase inhibitor which I discovered from my group, and I will discuss these 2 important leads like M873, which is extremely selective for alpha isoform. Just see the file. Alpha isoform IC50 is 40 nanomolar. But if you see the beta, gamma, delta, it is more than 20 micromolar. It is more than 500-fold selective for alpha isoform. So that is the beauty of this alpha, and that is the reason we wanted to pursue this in clinical studies, and now we are doing the all GLP tox studies, and we'll go for IND enabling studies now. Second compound which I will be speaking more about is a triple M290. Again, this is very potent inhibitor of CDK4, 6, and 9. We tested this in multiple cancer cell lines. We tested in animal models of the cancer. We did all transnational from this toxicology studies, even Ames assay, mutagenicity, reproductive toxicity, everything we did. Even we prepared the formulation and then we filed IND and we got the approval from the DCG also for this triple M290. And very soon we'll start the clinical trial of this M290 now. So to start with, I will speak about first PI3K inhibitors in 3-4 slides. So when this kinase inhibitor program was started, the 2001 was the year when first kinase inhibitor was approved. And during the same year, this PI3K drug discovery efforts were also started by the one of the institute called as Institute of Cancer Research in UK. They started one medchem program on this PI3K program, and they could come up with one of the candidate which is extremely potent inhibitor of PI3K alpha. It is a PI103. It is a pyridopyrimidine skeleton. And this scaffold is extremely potent inhibitor of PI3K alpha with IC50 of 3.6 nanomolar. But you can see it is inhibiting all isoforms of the PI3K. It is a pan PI3K inhibitor. And that is the reason if you your compound is inhibiting all 4 isoforms, you are liable to get the toxicity in clinical trials. And that is reported in multiple papers also. And another liability with this molecule is you see the structure, it is a kind of it is a planar structure. So it is well established in the literature, and it is based upon my own experience also. Structures which are planar structure, they have solubility concern. So if you want to improve the solubility of the molecule, bring in some flexible moieties in that. So water solubility is only 5 microgram per ml. So that is one of the concern of this molecule. And another third concern is it has very poor T-half. This OH present at the meta position undergoes glucuronidation very, very easily, and T-half you see, and in 10 minutes it gets metabolized up to 70%. So these are the 3 bottlenecks of this molecule. Although it has shown very good efficacy in the animal model, proof of concept was established in the animal model, but this molecule could not go to the clinical trials because of these issues. So this is the same institute, ICR UK, Institute of Cancer Research London. So they conducted a medical program on this molecule, and they could come up with 2 clinical candidates, pictilisib and apitolisib. These are the 2 candidates discovered by the same company, and these have better properties in terms of the solubility and some of the metabolic stability concerns. And both the molecules currently in the phase 2 clinical trial, and now this Genentech is conducting the phase 2 clinical trial of this molecule. For both the molecules. So this is how this discovery started with this PI103, and 2 candidates have gone to the clinical trial. So again, both these molecules are again pan PI3K inhibitors. So that concern was still not addressed. So we wanted to make some effort on this in this area. So we visited the crystal structure of PI3K alpha isoform and other PI3K isoforms also. And we identified that this GLN 859, this is the city which is more specific for the alpha isoform. And if you get some engagement with this receptor, glutamine 859, by some extrapolation of some of the linkers in this main pharmacophore, there is a chance to get the isoform selectivity advantage. So this was the criteria we put for the isoform selectivity. And another concern we wanted to address is this solubility concern. And we wanted to remove this phenolic moiety here. Phenolic moiety, we wanted to replace that with some saturated ring like piperidine, piperazine. So that these kind of molecules are flexible in nature. So if you see the 3D structure of piperidine or piperazine, they are not planar in nature. So that brings the flexibility, and because of that flexibility, we hypothesize that we'll get the improvement in the aqueous solubility. So using both these approaches, we wanted to design some of the compound, and particularly this pyrimidine morpholin, this skeleton particularly, it is very, very important for getting kinase inhibition. this pyrimidine, this and morpholin. This is important for binding to the ATP binding site, where this valine 851 is present. So we don't wanted to disturb any this main pharmacophore, and we wanted to disturb only this portion. So we planned a medicinal chemistry program. Almost 70 compounds we synthesized in this series to come to the final one compound. And this was one of the students, Shabir Mohammed. He was one of the most hardworking, and he synthesized 72 compounds in this series. And currently he's doing postdoc in United States in one of the university, Purdue University. So he started synthesizing this compound from this commercially available thiopine carboxylate ester. And at this position, we diversified into two different series. Like one series is the piperidine urea series, and another series was reductive amino series. So both the series we plan to synthesize. So although this amino alkyl series is not bringing some flexibility, but we wanted to, because this molecule also was showing the interaction with 859 residue. So all this, after synthesizing all this compound, our main objective was to screen this for PI3K alpha form inhibition. So I'm not showing the activity for all 72 compound, but I'll show you one important series where we got the hit compound. So this was our pathway. Like we tested all this compound against the single concentration of 500 nanomolar for alpha inhibition. You can see here, M873 is showing very potent inhibition, 74% inhibition at 0.5 micromolar. That is considered very good. And if you see the parallelly also, we did solubility studies, and you can see 800 microgram per ml is the solubility for this compound, whereas parent compound was having only 5 microgram per ml. So in terms of the both, we are getting the alpha isoform inhibition, as well as there is an improvement in the solubility. And this compound, then we study in detail. This is just comparison. I'm not showing in detail all this biology. So just see this table. The clinical candidates which are showing the inhibition of all 4 isoforms, whereas the 873 is highly selective for alpha isoform. So this is the one of the major advantages of 873. And it is improvement in the solubility, and PK also, IV PK currently we are done, and there is some improvement in the PK profiling also. We tested in animal model also of the murine model, and it is in vivo efficacy also we observed. Now our plan is to validate these results in human genome models. Now currently we have taken up this molecule for IND enabling studies under CSR Pan Cancer Mission. Now I will switch my gears to CDK program. So here we selected these two natural products, phascaplacin and rohitokin. Before I go to that, just briefly about how this CDK plays important role in the regulation of the cancer cells. So CDKs are key regulatory enzymes which are involved in the cell proliferation while regulating the cell cycle checkpoints and transcriptional events. You can see there are these blue colored boxes. All these are CDKs which are regulate our cell cycle. And in normal cells, whenever there is a DNA damage, it upregulates the expression of this p53, which is a natural suppressor of the tumor gene. So this p53 gets upregulated, and which eventually recruits or increases the levels of these natural inhibitors of the CDKs called as p15, p16, p18, p19. So levels of this get increased so that they can inhibit the CDK enzyme levels, because CDK enzymes are regulating the cell cycle. Ultimate aim, if there is a DNA damage or there is a faulty daughter cells are forming, you must stop the progression of the cell cycle so that multiple faulty daughter cells should not form, and their cancer cells will not get formed. So main aim is to inhibit the expression of CDK4 and 6. These are the main two players of this particular cell cycle. And so we can target CDK4 and 6. Even transcriptional CDKs also play very, very important, because they generate the translational mRNAs and RNAs required for the formation of daughter cells. And so these CDK7, and these are now people are targeting CDK7 and CDK9 also for getting anti-cancer agents. So till now there are these 3 CDK inhibitors currently approved by the FDA: palbociclib, ribociclib, and abemaciclib, which are selective inhibitors of CDK4 and 6. So this is the details which I mentioned when they are approved. Apart from these 3 FDA approved drugs, there are 3 orphan drugs also approved. Orphan drugs means the diseases where there is very less amount of population is there, maybe less. As per the definition of FDA, if there is a population of less than 2 lakhs is affected by this disease, which is called as orphan disease. And these diseases, there are not many pharmaceutical companies are putting their efforts. because less market opportunities are there, and that is the reason you get very quick approval. Even after doing the phase 2 clinical trial, you can get the approval. So these are the 3 candidates which are CDK inhibitor, which have got the approval from the FDA as an orphan drug, and one of that is a flavopiridal, which is the structural analog of the Rohitokin natural product. So first scaffold I will speak about, this is the fascaplycin. So this was discovered by Novartis in 1990s. It is isolated from the marine sponge called as a fascopinosis. So yield of this natural product from marine source is very, very low. You have to take the kgs or tons of the marine sponge, and you can get the milligram quantities of fascaplycin. So ultimately, the synthetic strategy is very, very important. So although there are 4 to 5 syntheses reported till date for fascoplycin, but since we wanted to work on this natural product, we wanted to first establish the total synthesis for natural product. And we develop a new process for fascoplycin. We synthesize this natural product in 2 steps, and we published this in Medchem in 2012. Then our another objective was to this natural product was having the liability of the unusual toxicity. That means it is showing the toxicity to the normal cells as well as cancer cells at the equal concentration. That means there is no therapeutic window. Although it is very potent inhibitor of the cancer cells, but there is no therapeutic window. That is the major bottleneck. It has not gone to the clinical trial. Lot of efforts have been taken by the NCI on this multiple panel of cancer cell lines and genome models. It was tested, but there is no selectivity. And that is the reason this molecule was dropped out and nothing has happened afterwards. So we started a Medchem program on this, and our objective here was to synthesize the non-planar analogs, because again, planarity is the one of the reason for DNA intercalation of the DNA, and because of the DNA intercalation, it is showing the unusual toxicity or no therapeutic window. So we synthesized almost 30 to 40 compound again here. 2 compounds which we identified where these are non-planar analogs and which we validated in the animal models, and in animal model they showed very excellent efficacy. Now, next molecule which we selected is a Rohitokin. So this natural product, this is connected to the diacetyl binding theorem. I'll speak briefly about this plant. So this is the plant which occurs in the India, and traditionally this is being used for the managing the inflammation, because this natural product was isolated in 1990s by a company called Host Life Sciences, which was located in Mulund, currently which is now Piramal Life Sciences. They, for the first time, they isolated this natural product, Rohitokin, and they reported this as a TNF-alpha inhibitor. That means an inhibitor of the inflammation. And based upon the traditional knowledge also, this compound was being used by traditional people. Some of the tribal people also in the Uttarakhand, they are using decoction of this plant for arthritic conditions. So there is a traditional information for arthritis. And major uses of this plant, because this is a huge tree, and the furniture of this tree was being used for the furniture trade also. But this plant is very rarely occurring, because as far as we found that this occurs in the Uttarakhand area and Western Ghats. But beauty of this natural product plant is that this contains a natural product called as a Rohitokin, and this is present in abundance. You take any part of the plant, you take a bark, you take a stem, bark, leaves, roots, any part of the plant, Rohitokin is present in good quantity. And this, see the quantity, 0.5 to 5%. Bark of the plant contains 5%. You can take 100 gram of the material, bark, and you can isolate 5 gram of the natural product. That is very, very good quantity. And this natural product has already delivered 2 clinical candidates. One is a flavopiridal and one is a rivasticlib. This is pyramal molecule. Both these molecules basically discovered by Indian chemists. That is another beauty. Flavopiridal was also discovered by Indian chemists, this Piramal Life Sciences. Later on, it was taken by the Sanofi Aventis. So discoveries happen in India, and both have gone to the clinical trial. So that was the reason this success has inspired to work on this natural product. So these are some of the points why we were interested in working on this natural product. So while we started working on this medicinal plant, these are the sub-objectives of this, like do the thorough phytochemical investigation, not only isolation of Rohitokin, but isolate other natural products which are also present in this plant, and do some medicinal chemistry, do animal biology, preclinical validation. All these are sub-objectives here. So as far as phytochemical investigation is concerned on this plant, we took each and every part of this plant. We separately collected, and we did phytochemical investigation of all the parts. Bark, we separately isolated natural product, leaves, even fruits and seeds also we separately isolated. These are the natural product, combined list of the natural product which we isolated from this plant. Rohitokin, obviously, we have observed that it is present in the leaves, bark, fruits, and stems. And apart from this, we isolated another natural product, Rohitokin N oxide, as a natural product. Then one of the regioisomer also we isolated of this rohitokin is a disolene. This was the first time we isolated from this, and this name we are given, that disolene is a name we given this to this compound. And then interestingly, this camptothecin and another derivative we isolated from this plant, and this is the new source of this plant for this natural product. These are for the first time we isolated camptothecin from this particular genus. And as an outcome of this phytochemical investigation, these are the publication from this phytochemistry work. And since rohitokin, although rohitokin has delivered the 2 clinical candidates, but rohitokin was never tested for its CDK inhibition and never tested for its anti-cancer activity. That was surprising to us. Although it has delivered the clinical candidate for flavopiridol, although it must have been tested, but literature was not available in public domain. So we tested this rohitokin for CDK9 inhibition, and it was observed that rohitokin itself inhibits the CDK9 with 300 nanomolar IC50. And when we tested in the panel of cancer cell lines, we observed that it shows the moderate inhibition of the cancer cell lines, IC50 around 10 to 20 micromolar. So that is not considered to be very good, but it is a good starting point for any cancer program. So we wanted to do the medicinal chemistry on rohitokin. So by using the computational drug design, because structure of the CDK9 was available, and so we did computational studies, and based upon the SAR properties of the flavopiridol, it was already established in the literature that these are the 3 important functionalities, like this OH and this functionality, this piperidine, OH, and N-methyl. These are 3 important functionalities which are important for the CDK inhibition. So we were never wanted to touch these functionalities, and these are the ACD4 functionalities where we plan our semi-synthetic studies. So these 4 series of compounds we synthesize, almost 70 compounds we synthesize, and this Srans Jain, he synthesized all these compounds. And all these compounds we tested for CDK9 and CDK2 inhibition, and also for anti-cancer activities. So if only for one of the series, I'm showing the data here. So one of the compound 11D, you can see here 2,6-dichloro compound. It is showing the inhibition of CDK9 with IC50 of 1.9 nanomolar IC50. This is very, very potent activity. And parallelly, it is showing this anti-proliferatory activity in the panel of cancer cell line with IC50 close to less than 1 micromolar. So this is one of the interesting observation we found. And parallel to that, we also observed that it is not showing cytotoxicity in the normal cell line, like IC50s, GI50s, 20 micromolar. That means 20-fold selectivity is there. So we decided to stop here lead optimization and focus on this particular compound. So we did the PK, a lot of ADME studies we did. So this is just a brief summary, whatever studies we did, like all experimental solubility, log-P studies, metabolic stability, we tested this compound in a panel of CDKs. Even we tested this compound in a panel of 468 kinases, because kinase profiling is very, very important in cancer domain. We tested this compound and to see whether any liabilities is there. Then panel of cancer cell line we tested in order to select the proper for the development. So we observed that leukemia and pancreatic cancer, these are the 2 types of cancer where we can develop our compound, because this 2 type of cancer compound is very, very potent. Then as a part of the regulatory studies, we did CP inhibition studies, because our compound should not inhibit these 5 regulatory CYPs, 1A2, 2C9, 2C19, 2D6, and 3A4. So here our IC50 was more than 20 micromolar. That was also another good thing. So then another point was we tested for the Cacao permeability. This is another important point, because if your efflux ratio is more than 2, that means your compound is a substrate for Cacao efflux pumps, and it is likely that your compound will get efflux out of the blood by this efflux pump protein called as PGP. So if our efflux ratio was 1.8, that means it is not a substrate for PGP. So there is no liability of efflux pump substrate. Then plasma protein binding, metabolic stability in liver microsomes of even human, monkey, mouse, human. All this we did as per the regulatory guidelines. Even MET-ID also we did, and we identified that there are the 2 important metabolites: ND-methylation. So there is an ND-methylation occurs, and hydroxylation of this aromatic ring. These are the 2 major metabolites are forming. And we did detailed PK studies, even pancreas, how much it is reaching to the pancreas, because we wanted to take this to the pancreatic cancer. So we check its exposure in the pancreas, how much concentration of the compound is going to the pancreas. And then we validated this result in the human genomic models of the 4 types of the cancer. Blood cancer, this is a MOLD4, MIAPACA2 is a pancreatic cancer, SCT116 is a colon cancer, and SW620 also colon cancer. So all 4 types of the cancer compound is showing very good efficacy. And this whole work, preclinical pharmacology, we published in JMedChem 2018. So further details, so whatever I discuss, I think I discuss only up to in vitro and in vivo studies. And most of the medical chemists or academic researchers stops at this point. In vitro, in vivo studies, publish and forget it. So these are the tricky parts which I think nobody wants to go ahead because a lot of finances are required, funding is required. So we pursued this molecule and based upon the CSR funding, we got a lot of funding from CSR to conduct all these studies. So ADME pre-formulation DMP studies we did on this molecule. So I'll not be showing much details here because that we are doing the clinical trial. So I'll not showcase here. Then we did acute toxicity in 2 rodent species as per the DCGI guidelines. Then repeat dose toxicity in the 2 rodent species. Mutagenicity Ames assay we did the cardiotoxicity, this hERG channel assay, whether compound is binding to the hERG channel or not. And male fertility study also we did. And based upon these toxicity studies, we had to calculate the human equivalent dose. So based upon this, we calculated that 4 milligram per kg as our human equivalent dose. And based upon that, we prepared a oral formulation for this. Because based upon this HAD, we prepared a capsule formulation which contains 90 milligram of 290. And then as a part of the IND dossier, we have to prepare the phase 1 protocol also, because IND submission, basically it is intended to get the permission for phase 1 clinical trial. So you have to propose what kind of trial you are looking for, how many patients will be there, what are your endpoints, what are your objectives. So all those details we have to provide as a part of the IND dossier. So we prepared a phase 1 protocol with the help of one of the oncologists from the Bangalore. And this is the briefly about the clinical trial protocol, where our main intention, this is the first in human clinical trial. And since this oncology candidate will not go for the healthy volunteers, it will be this kind of clinical trials will be done in the actual patients, pancreatic cancer patients. Our main objective here is to do the exposure in the cancer patients. So primary endpoints are MTD, dose limiting toxicities, adverse effects, severe adverse effects, and also the exposure. And extrapolated objective is the disease control rate and some of the markers, key markers of the pancreatic cancer also we'll look for. And this study will be done in very small group of population because this ethical guidance does not allow to do phase 1 in multiple cancer patients. Only 12 patients are allowed by DCGI to us. And last year we got the DCGI grant for conducting this phase 1 clinical trial in metastatic pancreatic cancer patient. So because of the COVID region, since last 1 year, we could not start this clinical trial, but very recently we got funding from the CSIR. Now we are making the GMP formulation for this 290 so that within next 2-3 months, we can initiate the phase 1 clinical trial in cancer patients. So this is the journey of triple and 290. The journey started with the selection of the target, like CDK target we selected. Then we selected one of the scaffold for lead optimization. This we started in 2012. And this is how we reached to the clinical trial. So most of the hurdle, like after the 6th milestone, these are the major objectives. Most of the studies are done by outsourcing. All these studies are done by the CROs, which are located in Hyderabad, Bangalore, and we paid to them and they did studies for us as per the regulatory guidelines. And after getting the IND approved, now we are doing the clinical trials. Now, second plant, which I'll be speaking about, first plant, success story of the first plant I completed now, Crocus sativus. So this is basically for the Alzheimer disease. So briefly, I'll speak about how we started working on Crocus sativus in Alzheimer domain. So briefly about Alzheimer disease, this is one of the incurable neurodegenerative disease, and this is the background. I will not speak about this. So if you look at the disease, this is kind of a unmet medical need. So whatever available in the market, there are only 4 drugs are available in the market right now. Donepezil, rivastigmine, galantamine, and memantine. And all these are providing only symptomatic treatment, whereas the progression of the disease cannot be stopped. This is one of the problems of Alzheimer's disease. Very recently, in 2021, one of the monoclonal antibody was also approved by the FDA, but there are a lot of question marks like whether it is really efficacious or not. There are some of the issues. So again, small molecule is not there for the treatment of Alzheimer's disease. So we wanted to make some effort on this. So if you look at the normal versus Alzheimer brain, so there are the 2 main pathophysiologies in Alzheimer disease. One is a deposition of amyloid beta plaques in between the neurons, the extracellular in between neurons, synaptic clefts. So these are and inside the neuron, there is a formation of neurofibrillary tangles by the tau protein. So these are the 2 pathologies which are present in the Alzheimer brain. And because of this deposition, neurotransmission gets halted and there is a memory loss and so many things happens. So major effort which were put by the drug discovery or multinational companies are on these 2 pathologies, how to stop this formation of amyloid beta plaques, how to clear this A beta plaques. So in 2010, when we started working on this area, there was two independent studies, one from the Netherlands and one from the University of Washington. They published these two papers, which are based upon the clinical trials. They observed that there is a decreased clearance of A beta plaques from the brain. That is the primary concern. It is not the deposition, but it is the decreased clearance. And why that, how that is happening? That there in Alzheimer brain there is a overexpression, down regulation of PGP protein. This is the protein which takes out the A beta from the brain to the blood circulation, and that's how it gets eliminated. And if there is a decreased regulation of PGP, so we thought that why not to induce the expression of PGP by small molecules. So we wanted to induce the expression by upregulation of PXR. These are the nuclear receptor which can regulate the PGP expression. So based upon this hypothesis, we started a medchem program, and we selected almost 4 to 5 scaffold. We synthesized more than 200 compound, 250 compound in this. We established in-house assay for the PGP induction. And one of the students, this Sudhakar Manda, he synthesized work on 3-4 scaffold, and a library of compound was tested. And in each series, we identified some of the lead compound, and one of the compound we validated in animal models also of the Alzheimer disease. And from this, all these efforts, which I briefly summarized in a single slide. So this is the molecule which came from the natural sources of crocin-1. It's a carotenoid compound. We at the terminal end, there are sugar moieties attached. This compound has showed very exciting activity. This is the structure of that trans-4-JG crocin. So both the moiety, there is a Gen-Shiba sugar attached. And this compound then we validated in animal model like in transgenic mice model. This is the model 5XFAD, and this we did with the collaboration with one of the US collaborator in University of Louisiana. And this compound at 10 milligram per kg, it is clearing the A beta from the A beta Alzheimer brain. So this was one of the interesting fact that, and when we did the prepared the extract of this saffron, we observed that crocin-1 is the major compound. This is the peak for the crocin-1. It is present in 36% weight by weight in this crocin saffron extract. And these other compounds are also other crocins, which are just varied by the type of the sugar attached to this crocin, glycon part. Then we wanted to explore this saffron extract itself because it is crocin-1 is present in the major quantity. So we pursued further. We collected a saffron from the market. Then we prepared the crocin-rich extract. Then this extract itself we tested in multiple in vitro and in vivo assays. In vitro, like we check the compound effect of this saffron extract on the blood-brain barrier integrity, whether it induces the expression of A beta clearance, so neuroprotection, whether it protects the neuronal cell lines, whether it shows the neural inflammation. So all these studies we did, we validated in animal model, and we observed that extract is showing very, very excellent activity at 50 milligram per kg. It is showing the multi-targeted effect, like it increases the clearance of A beta. It is showing the anti-inflammatory effect, anti-neuroinflammatory effect. Even it shows the antioxidant activity, multiple activity it is showing. And this work we published in ACS Chemical Neuroscience. And then later on, we thought that since this parent compound, if you see the structure, it is a simple ester moiety, and it is obvious that it will not go to the brain plasma circulation because of the liability of the hydrolysis. And so whatever goes to the blood circulation or even whatever goes to the brain is actually the glycan part, which is called as a crocin. And this we observed in the PK studies also, pharmacokinetic studies. When we administered crocin by oral route, whatever was observed in the plasma is a crocin. And even in the brain also crocin was there. Crocin was not reaching there. So we synthesized crocin, and we tested this crocin also in vitro and in vivo assays, and we observed that crocin is showing excellent efficacy. And very recently, this work also on the pure molecule we published in the autophagy very recently. So this is the background or science behind our saffron-based product. Then what about the technology? How we can outlicense this product? Because a lot of science and interesting facts are there as far as the Alzheimer disease is concerned, because we validated this knowledge in the transgenic mice model. So to translate this technology in the form of a technology, we prepared an oral formulation of this. And since we are planning to outlicense this, we need a patent on this also. And getting a patent on any natural extract is the next impossible job. So for that purpose, we prepared a novel formulation, sustained release formulation. And because of this formulation, we increase the exposure of crocin by 4-fold compared with the plain extract. So this was one of the inventive steps which we observed, and because of this, we got IP on this, and this IP is also granted in one or two countries till now. And in PK studies also, this crocin, you see this here, this gets converted to the trans crocin. So and this work we published in ACS Omega. And in 2018, this technology of a nutraceutical product we out-licensed to one of the company from the Gujarat, Farmanza Herbals, with the brand name of Saffron Tea. And not since this is nutraceutical was a short path, but since we have the data on this animal model also, now currently we are doing the regulatory toxicology studies. We have the toxicity study, but in non-GLP studies, we are doing now GLP studies so that we can file an IND application to the DCGI, so we can start the clinical trial in Alzheimer's disease or in dementia by phytopharmaceutical DCGI route. So that currently we are doing now. So as a part of Alzheimer's disease also, since it is, I was mostly talking about the plant extract. So just in one slide, I'm mentioning how we are again connecting to the discovery of new leads for the Alzheimer. So in my group itself, I always plan to maintain the library of the pure extract, library of the pure compounds. So whatever compounds students synthesize, whatever extract students prepare, I deposit here. And so almost I have 150 extracts. So all those extracts I tested for different targets of the Alzheimer's disease. So all these targets we established ourselves, and we observed that out of that, 2 extracts have shown the multi-targeted effect. We did in Pampa BBB assay, like whether it permeates the membrane, CNS membrane. And then we did in vitro and in vivo studies also. We validated in the animal model also in the dementia. And recently we published this plant extract work in the phytomedicine. And also similarly, like plant extract, we tested our own natural pot library. Almost 150 natural pots are there with me. And we tested against all these 3 targets. And these are some of the interesting hits we observed. And now we are doing the medicinal chemistry on this natural product. And some of the work publications which came out of this screening work these are some of the publications I listed here. Now, third plant is a Burgeonia accelerator. This is the last plant, and next 3-4 slides, I'll finish my presentation. So this is a Burgeonia accelerator. So why we started working on this plant? This plant occurs in the temperate region of Himalayas, and this plant is already reported in the Ayurveda for multiple diseases. Like mainly, it is used for the kidney stones. So when we started working on this plant, we isolated Burgeonia in very good quantity. This is the natural product which is present in abundance. It is a unique class of natural product, C-glycoside, present in this Burgeonia accelerator. And first, once we isolated Burgeonia, we synthesized a library of compounds. We wanted to do the medichem on this product to get a feel on this natural product, its stability, even whether it's animal efficacy, all that feel we wanted before we switch our gears to the phytopharmaceutical. We synthesized almost 18 to 19 compound, one series of compound we synthesized, and all we tested against TNF alpha and IL-6 cytokines. And it was observed that this TRIPM17 as well as Burgeonia, it has shown the selective IL-6 inhibition, and it is not inhibiting TNF alpha. That is one of the interesting observations we observed, because nowadays if somebody is working in the TNF alpha IL-6 cytokine area, nobody wants to touch TNF alpha, because TNF alpha has certain liabilities, like there is a black box warning for TNF alpha antibodies. Like if you take the TNF alpha antibodies for arthritis, there are chances of getting tuberculosis infection or HIV infection. So people are always preferring selective inhibition of IL-6. So not only IL-6 inhibition, but this also shows the anti-pain effect or anti-nociceptive effect. That means it is a dual effect of IL-6 inhibition and anti-pain. So this is one of the reason we wanted to prepare the Burgeonia extract and develop it as a phytopharmaceutical lead for management of pain in rheumatoid arthritis. And that's how we started working on this Burgeonia extract. And we prepared this extract. We standardized with 4 markers as per the DCGI guidelines. And Burgeonia is present in very abundance, like 9% is Burgeonia in this extract. We completed CMC documentation and in my validation, everything we completed. We got IP on this. Even we completed regulatory toxicology, safety studies we completed. And last year we filed IND application to the DCGI now. And when they came, they have asked us to do one more study. Like we did not do the non-rodent study because that was highly expensive. Non-rodent study means you have to do the safety study in dog or monkeys. So if you go for the dog studies, it cost at least 1 crore. And that much funding was not available. But we could not convince DCGI that this plant is present in Ayurveda. It is people are consuming since long time. But still they wanted that we should do the non-rodent safety. So now we are doing the non-rodent safety so that we can start the phase 1 clinical trial in healthy volunteers. Another phytopharmaceutical lead is a Rohitokin enriched disulfide bioactive. This currently we are doing the IND enabling studies. So this is the briefly about the Burgeonia extract. We prepared a Burgeonia extract. We validated in vitro assays, assays of the inflammation as well as the pain models. And Burgeonia itself we validated in animal and pure molecule itself in vitro and animal models. And this is the formulation we prepared, gastroretentive sustained release formulation. So if you see the structure of Berginin, I think you've seen this is a lactone moiety is there. So this moiety has some concerns like it gets opened in the intestinal pH. So that was one of the liability of Berginin, and this has given us an advantage that we can make some kind of a new formulation, and we prepared a gastroretentive formulation. That means this formulation, these capsules, once they go to the stomach, it has a buoyancy effect because of the swelling of the capsule, and it does not cross the pyloric sphincter and does not go to the intestine. So whatever absorption occurs of the Berginin occurs from the stomach. So because of that reason, the plasma exposure of Berginin was improved by 4-fold in comparison with the plain extract. And based upon this, we got a patent on this extract. And this work we published in this journal in last two years back. And very recently, in October 2021, this technology we out-licensed to the Viridis Biopharma from Mumbai as a nutraceutical product, because since company was interested, we don't wanted to stop here. Although we're pursuing this product in a phytopharmaceutical route, but we out-licensed this technology in a nutraceutical mode. So they are now planning to launch this product in the market for rheumatoid arthritis. Now, this is just summary of my whatever contribution to the science in last 10 years. I'll not speak much about this. So most of the part also I did not present here, like some of the effort on the synthetic methodology I did not present here. So this is the slide which I showed very early in my presentation. So we started working on these 3 plants. So what is the outcome from these 3 plants? So from Diacetyl Banactiferum, we could discover 2 leads. M290, which has gone to the phase 2 clinical trial, which is a semi-synthetic derivative of a natural pot isolated from this plant. M64, it is a phytopharmaceutical lead, and currently we are doing IND enabling. Bergenia Acetylata, we out-licensed product to the industry, and one product of this M160, we are doing the phytopharmaceutical IND. We filed IND application. And in Crocus, we out-licensed the technology as a nutraceutical, and currently we are developing it as a phytopharmaceutical lead. So this is the outcome of these 3 plants. So this is my current group. Currently 7 students are doing PhD with me, and 1 postdoc is also with me. And these are the 11 students which I graduated from my group. So this is the list of students. Just I wanted to show here. So these students, which 6 students work in the area of oncology, and this 5 students work in the area of Alzheimer disease. And all of these students are very well placed now currently. Just I'll show you here. Like Shreyansh, he's assistant professor in IIT BHU. And one of my first student, he's doing postdoc in Monash University, Australia. Baljinder Singh, he's scientist in AstraZeneca, USA. This student, he has completed postdoc in US and now returned to India, and he's working in Sci Life Sciences. Then this Shabbir, he has done postdoc from Taiwan. Now he's doing his 2nd postdoc in Ohio State University. Rani Sharma, who has last 2 years back, she completed PhD. She's RA in DRDO, Chandigarh. Then Sudhakar Manda, he completed his postdoc from South Korea. Now he's working as scientist in Origin, Bangalore. Rohit Sharma, he has completed PhD. He worked for a short time in Ireland, and now he's got permanent position in State University in Himachal Pradesh. Prashant Joshi, he is working as a faculty member in NMIS Chitpur. This Abu Bakar Wani, he is doing postdoc in Washington University. And recently, this 2 months back, this student has completed PhD, Muhammad Abdullah. He has got recently postdoc in Canada. So this is, I think all students have worked hard and they got the good positions in various places. And this was the picture which I wanted to share. Like, this was the first moment when my first student defended his PhD in February 2015. Ramesh Mududola, one of the hardworking students, and almost published 8 to 9 papers. And he's one of the good students, I think. So, finally, acknowledgement, my biology partners, outsourcing partner, most of the IND enabling studies are done, kinase profiling done at the University of Dundee, Dabur, then Jubilant, Eurofin Biosys. All these companies have done all safety pharmacology. Obviously, we pay to them, but these are the players we played a very important role in our IND filing. And these are my other collaborators. And funding agencies like DST, SERV, DBT, CSR Scientist Grant, CSR FTT Grant for my IND enabling studies for 290, and Phytomission project. And most importantly, both the directors, previous director Ram Vishwakarma and Dr. current director DS Reddy, I must thank them. Thank you very much. And finally, I should acknowledge my alma mater, Department of Natural Product, because whatever I'm doing or whatever I'm today, because of this basic learning which I got from the NIPER. Because before I joined NIPER, I was not knowing anything about the research. So whatever I learned, this isolation, what is natural product, how we can do the NMR characterization, everything I learned in NIPER. So during my 6 to 7 year team, 2001 to 2007. So I must thank my teachers, Professor Indapal Singh, Dr. Sanjay Jachak, late Professor K.K. Bhutani, Dr. Arvind Saklani, who was the faculty that time, and other faculties also from other departments, because they taught me on different subjects. All the director, Dr. C.L. Cole, Dr. H.P.S. Chawla, P. Ramarao, Dr. K.K. Bhutani, all my department seniors, D.D. Singh, Rahul Pawar, Hemant Jadhav, Atul Jadhav, Selvam, Chitra Ji, Aruna, Yatin Shukla, all these are my immediate seniors. And my colleagues, Atish, Aji, and Nana Oli. And my departmental staff like Dr. Balka Singh, Anwa Singh. I think I just recently I saw that she has also retired now, Anwa Singh, Dr. Sandhu, Dr. Tripathi, Prasanna, Sanjay, Amit, Rakesh, and Mohammad Shahid. Thank you very much.
Yeah, Sandeep,
Yes, sir.
hello. Yeah, very nice to see you and listen to you. I think we have also Siddesh, Raju, and one more, I think. Atul, yeah, all there. So nice to see you all on this. I think it's a good and great occasion to be here with all of you. And I think we had excellent presentation. Sandeep has been our speaker in our DD NPTM conferences since 2012. So I think we have heard him several times, and again today was another very good lecture experience to hear him and share the knowledge. And what I feel, Sandeep, no, whatever you have shown, like starting with phytochemistry, isolation, characterization, medicinal chemistry, and very important, as you mentioned, translational research, converting your research into a product. This is very important in today's context, as we are actually every funding agency. Wherever you go, they ask this, what will be the outcome? What is the product? And it is very inspirational to all our students. I guess, no, we have a lot of PhD students now, so they need to take a very good lesson from this. Okay, how the work, whatever you are doing, can be translated, can have commercial application, which I think Sandeep has very nicely demonstrated. I think it was wonderful, Sandeep. And I have one question only regarding with respect to saffron. No? You said the extract was equally active or even better active than the quercetin, or let us say crocin. But then why actually you have shown the work with the quercetin? You are taking this as a phytopharma, as if I understand the extract?
Yes, as a phytopharmaceutic, yes.
So you are not taking anything lead molecule from quercetin or something?
No, we are not going in NC route. NC route, we need IP on the pure molecule. We don't have IP on the pure molecule. We have the IP on the phytopharmaceutical lead. And crocin extract, it is actually effective at the 50 milligram per kg dose, whereas the pure natural product crocin, when it is effective at the 10 milligram per kg dose, because of the content of the crocin.
Right, but what about the stability of crocin?
Crocin, it is obvious that it will not be stable, but we proven that crocin gets converted to the quercetin, and quercetin is actually the bioactive marker. It is a kind of a prodrug quercetin.
Exactly, yeah, that is there. But what about its physical stability, light stability, photostability? This compound is liable to photosensitive as well.
Yeah, it is a
I think there must be a challenge also handling.
Because it is a carotenoid, multiple double bonds are there. Even it undergoes the isomerization, double bond cis trans isomerization also happens.
Yes, that's true. Right. So I think it's very wonderful and great presentation. All the students, session is open, I think, for question answer, and you can ask him some questions. All our PhD students are there, I guess. So it is very nice to see you again, Sandeep. And thank you for accepting this invitation and sharing your knowledge, because I know you had a lot of data, but you managed to show it in 50 minutes or so.
Yes.
It was wonderful effort. Yeah. Okay, all the best.
Thank you, sir.
Sure. I think if anyone has questions, Sandeep, we can have few questions.
Yes, sir. Sure. Yes, sir.
Sandeep, Chideshwar here.
Yes.
Yeah. I don't have any question, but like as you are our senior and you are like a role model for us. I recently started my career in academics, like last 1 year, and seeing your work, I'm really motivated. The kind of volume of the work you have done, and actually the translation work particularly. I'm really very impressed with the way the work we have done. I'm very fortunate to, it's like to have a senior like you. Thank you very much.
Yeah. Thank you, Siddheshwar. So there is one question in the chat box. Just I will answer that. Vilas Jagtap is asking how name was given to the compound, newly isolated compound. Just I will briefly answer that. So it is up to us, but always we have to stick to some guidelines, like whether follow something genus name or species name, something we have to consider, and some class name also. Like we isolated that compound, we given name as a disolene. So diso comes from the species name, like genus name disoxyxylum, and ion is for the alkaloid. So that by that criteria we given the name disolene.
Sandeep, I have one more question actually. Actually, the phytopharmaceutical that you are planning to launch. So for phytopharmaceutical, we need to have a standardization with minimum 4 markers, right?
Yes.
Yeah. So do we need to perform the clinical trial also through India route, or like how is the actual approval process?
So clinical trial, everything is exactly similar to the small molecule, new chemical entities, small molecules. So regulatory agency is also same. DCGI is regulating this phytopharmaceutical. All safety studies, clinical trial exactly as per the clinical trial guidelines of small molecules.
But since this crocin has a traditional use, so you can get exemption, right, on clinical trials?
No, they are not allowing that. They are very stringent on this. Even US FDA, US botanicals, they have some. They are more liberal. If there is already human data is there, even they are not asking for the animal toxicology. But our DCJ is asking everything.
Okay.
So that is the kind of I think trouble we are facing in here.
Okay. So are you planning to launch these nutraceutical products particularly? Like you are out licensing this product, right?
Already out license.
Out license. Yeah, already out license.
Yes.
Okay.
Sandeep, Atul here.
Atul.
Great work. I mean, I'm really feeling very proud to see all such a humongous work which you have done. Great work. So, just to add to this youngsters there, I would just like to highlight one important thing which might have got missed in the brief. Sandeep was the only natural product chemist got selected in the Reddy's campus. I mean, they came for recruiting medicinal chemists, and I still remember very well that he was the natural product chemist coming from our natural product department, getting into Dr. Reddy's medicinal chemistry division. So that was one of the achievement. I mean, he's been an achiever, no doubt. Everywhere he has been making a name, and we get to hear his name somewhere always. So it's a great thing. I mean, we are all fortunate, rather Siddheshwar, you, me, to be associated with these great people like Rahul, Sandeep, Hemant, Chitra. So these are a great scientists we had association. So Sandeep, just on a passing thing, because I heard you that you had a trouble to get DCGI. So being a government organization, having a government project, I mean, I think you were better placed to negotiate some of the people, right? I mean, I'm still surprised that you are having trouble getting things cleared off.
No, that those kind of efforts now we are putting forward. Even from CSR side, now CSR has created one a committee where we can do some flexible changes in phytopharmaceutical guidelines. Like if there is a human data is available, even this 4 marker criteria also people are not comfortable. Enriched fraction also people are thinking that enriched fraction is not really required. If USFDA does not say anything about enriched fraction, even you extract anything, we can file a US botanical. So those are getting changed now. We are doing those efforts. CSR, DGC created some committee task force. So by that task force, I'm also part of that task force. Now we are changing those guidelines. At least efforts are being made.
So how are you handling your screening technologies? Like you are having some research scientists employed with you so as to get the enzyme screening or these things done?
No, enzyme screening we do. My students do. All these in vitro assays we do. I have my instruments, plate reader, my multimode reader, everything is there.
Okay. Now, there is another important chain actually. If you see the chain of events, Dr. Bhutani came from RRL
Yeah,
Jammu
yes, same. Yes, yes.
and started the department, and you are the one a DNP produce which has joined Triple IMs for RRL Jammu basically.
Yes.
So it's completing a cycle actually.
Yes, yes.
Okay. Thanks. All the best. Thank you so much for having this platform.
Yeah, thank you, Atul. Sir, you are on mute.
Okay. So thank you, Dr. Atul. Yeah, Dr. Sandeep, there are a couple of more questions for you in the chat box.
Okay, sir. Okay, sir.
So one is from Mr. Vilas Jagtap, which things to consider at the time of translating lead into phytopharmaceutical. And the other question is how to solve low aqueous solubility of a molecule.
Yes, I will answer those.
Yeah.
So first question was what kind of parameter we should consider whether that we should translate to phytopharmaceutical. The first thing is that since we do the phytochemical investigation and you get a feel on that particular plant, what kind of secondary metabolites are there, whether that plant contains secondary metabolites, because the important criteria is that we need four markers. So whether those four markers are present in your extract and whether those markers are stable. So those kind of things. And although it is not mandatory that it should be bioactive marker, but one of that should be bioactive marker, then only we can actually correlate your extract with the pharmacological activity. So in PK, whenever you do PK of the phytopharmaceutical lead, you have to monitor that particular bioactive marker, whether it goes to the blood circulation or not. So ultimately, because then only you can correlate your efficacy. So these are some of the parameters we should consider for translating that initial extract to the phytopharmaceutical lead. Second question is how we can solve the aqueous solubility concern. So aqueous solubility, generally this kind of concern comes with the lipophilicity, because if molecule is lipophilic, if it is a rigid compound, all rigid compounds are very poorly soluble. So planarity is one of the rigidity example of rigidity. So if you just break the planarity, break the rigidity, bring in some flexible moieties, you overcome the solubility concern, you can get the solubility. So these are some of the some polar functionalities we can introduce. So those kind of small modification we can do to improve the solubility. Yeah, this is
yeah. Thank you, Dr. Sandeep. Any other questions?
Hello, good afternoon, sir.
Yeah, Ankur.
Yes, sir. This is Ankur, sir. Sir, I have one question. Sir, did you find any other compound than crocin-1 which has an decreased clearance of amyloid beta from the brain in the crocus sativa?
So if you look at the phytochemical constituents of the crocus sativas, 60 to 70% are crocins. There is nothing is available other than crocins, because this is a stigma. This is not a leaves or bark or something like that. Stigma is orange in color, and whatever crocins are orange in color. Everything, it is full of crocins. There are 16 crocins reported in saffron, and all are containing same glycan part, which are differing only by sugar moieties. So all 16 crocins get ultimately hydrolyzed to one single crocitin. So we did not bother about which crocin is effective, because ultimately crocitin is effective. All 16 crocins are products for crocitin. So we did not test other crocins because it is very difficult to isolate crocin, all crocin pure natural product, and because they are not stable. If you only isolate, they get degraded after some time. That is another concern for crocins.
Yes, sir. Thank you, sir.
Okay.
Okay, I think we have to stop somewhere. Okay. And we have had some questions. We had some of our alumni present, and I can see Purvi also is present. Hello, Purvi.
Okay.
Hello. How are you, Purvi?
Good to see you all. Good to see you all together, sir. And very nice presentation, Sandeep, sir.
Yeah, thank you, Purvi. I never met you, I think, but yeah, thank you.
Okay.
Yeah. So I think it was a great presentation, and you have done some outstanding work, Sandeep, and we are all very proud of you, what you are doing there at IIM. All your seniors here and both your teachers are very proud of you. Good luck for your future also.
So thank you very much for the invitation. So
Yeah.
thank you very much.
So, Debanjan or Monica, please continue.
Yes, sir.
Yeah.
Now I would like to ask first year PhD student Yogita ma'am to give the vote of thanks.
If you go, then you can catch Siddheshwar now. We have already got him on the platform.
Yes, sir. Yes, sir.
So next,
That is our in pipeline.
next speaker you can.
We will plan.
Yeah.
Yes, sir.
Okay.
Planning for this.
Thank you.
Okay. Thanks.
Thank you, Atul sir. We will definitely do some good work.
Yes, sir. Yes, sir. Thank you, sir.
Thank you, Monica, for this wonderful opportunity. Now that we have come to the end of today's webinar session, I would like to offer a special thanks to Dr. Sandeep Bharate, sir, being Principal Scientist at IIM Jammu, even in his busy schedule, for spending his precious time with us and guiding us with this informative session. Thank you so much, sir. I would like to thank our Head of Department, Dr. Sanjay Jachak, sir, and Professor Dr. Inderpal Singh, sir, for the constant support to make this webinar in a reality. Thank you to our scientists, faculties, Dr. Alok Goyal, sir, and Dr. Tripathi, sir, for their precious time. Thank you to our technical staff, Mr. Sanjay Veer, sir, Mr. Mohammad Khan, sir, Mr. Amit Srivastav, sir, and junior technical staff, Mr. K. Prasanna, sir, Mr. Rakesh Kumar, sir, for their gracious presence. I would like to extend my thanks to Dr. Desh Deepak Singh, sir, Dr. Atul Jadhav, sir, Dr. Siddheshwar, sir, Dr. Raju, sir. Also, I would like to thank our PhDs, placement coordinators, my batchmates, and all for making this session successful with their presence. Once again, I would like to thank Dr. Sanjay Jachak, sir, and Dr. Inderpal Singh, sir, for their endorsement, without which this webinar would not be possible. Thank you all for participating and making this webinar successful. Lastly, I would like to remind all the participants to fill the feedback form, for which link is mentioned in the Google Meet. Thank you all. Have a nice day.
Thank you all.
Thank you.
Thank you, everyone.
Thank you, everyone.
Sir, I have just one request that can we on our front camera just take a screenshot or just take some screenshot. ले
और तो उसको एकदम क्लीन एनवायरनमेंट चाहिए तो एक दिखाई है बीमार अभी तक इसका एक हमारा कंपाउंड करने वाला
Thank you, sir. Sir, this is just our LinkedIn page, and our page is reaching higher and higher views, and that is why, sir, we are doing this, and we are posting and we are updating our page in regular basis and periodical basis. So that is. Thank you, sir.
Great job, team. Great job.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you.
Thank you.